Thank you. So, hi everyone. My name is Sara Fumagalli. I'm a PhD student at the University of Milano Bicocca, and today I'm thrilled to present you the workshop that we have recently organized, which is called Data Hunters Workshop. So, in recent years, the rapid advancement of high-throughput technologies has generated a huge amount of biological data, blurring the lines between biologists and computational scientists. Life sciences research has thus become highly computational, necessitating professionals who have both biological and computational skills. For this reason, ensuring access to the bioinformatics field shall be on the agenda of life science degrees. This especially applies to biology-related degrees, where future biologists often ignore its existence due to the scarcity of dedicated courses. One of those highly computational fields that is also increasingly becoming data-driven is metagenomics. The vast amount of data generated over the past 15 years is currently stored in public international repositories, representing a valuable resource. Indeed, in the last years, metagenomics has moved towards integrating multiple data sets to capture the full complexity of its samples. However, retrieving and reusing these data sets is the main challenge for the majority of the researchers due to the lack of metadata standardization and low adherence to common ontologies. Indeed, before using these data sets, extensive and laborious steps of manual curation are necessary. For this reason, the current trend is to build specific collection of microbiome metadata. Indeed, these collections contain curated metadata which are readily usable, adhering so to the FAIR principle: fundability, accessibility, interoperability, and reusability. Continuing our efforts started in the 2021 with the Skin collection, which is a collection of human skin microbiome metadata, we have been working on improved version. Indeed, we have implemented an enhanced pipeline for projects retrieval and, of course, keep it updated. However, creating a collection is highly time-consuming and requires a significant numbers of people to achieve this monumental task. Interestingly, current curated metadata for a collection not only gives the opportunity to deepen the knowledge in metagenomics, but also offers valuable skills in data manipulation, representing an ideal scenario to familiarize with a programming language, for example, Python, and develop a solid understanding of this language. For this reason, we organize the Data Hunters Workshop as a fusion of our two aims, which are education and scientific research. Data Hunters is a learn and play activity of student science, custom tailored for the students of the University of Milano Bicocca. It was designed to equip students with skills for one of the roles of the bioinformatician behind the scenes of the microbiome field, like the metadata curation. It's more than just a learning experience, it's a proactive step towards addressing metadata standardization challenges in metagenomics. Focusing on biology students, we introduced Data Hunters Workshop for the first time at the Department Day of Biotechnology and Bioscience Department at the University of Milano Bicocca, and where we launched workshop registration on that occasion. And of course, then we promoted the workshop using our social media channels. The workshop was structured in 4 phases, which I present you in the next slides. So, starting from the beginning, the workshop kicked off on the 28th of February 2024 with a 6-hour frontal lecture. In the morning, we conducted a theoretical session on metagenomics, where we covered the fundamentals such as biological sample information and technical details. This theoretical knowledge was essential for the practical phase that followed, where we asked students to analyze selected papers and find key values that we discussed at the end of this practical session. This hands-on exercise simulated the task they would undertake in the following ways. In the afternoon, then, we moved to the bioinformatics lesson, which was organized, again, combining theoretical and hands-on practice. We introduced the terminal and basic bash commands, followed by an introduction to Python. In the second phase of our workshop, we introduced an autonomous hands-on experience. Here, the students access to two built-in resources that we develop. The first one resource that I present you is the educational resource developed on GitHub, which has enabled students to learn Python basics. As you can see from this screenshot, each theoretical section on data frame manipulation was followed by ad hoc exercise and the corresponding solutions. Except for the titles that you can read are in English, the rest of the resource and all the other resources that we provided during the workshop were in Italian to facilitate the student learning. The second research that I present you is the command line script built in Python. This research was developed to guide students through the metadata curation protocol. Designed in a playful style, the research allows students to follow the necessary steps for metadata curation and they assess the commands they needed for each step. Indeed, for each step, they could require hints if they forgot the commands to use, or also view the solution showing the correct command to use. Moreover, the resource was built to let participants to choose the level of Python they wanted to use, tailoring the experience to their skill level. Thanks to these resources, students have then stepped into the roles of bioinformatician, actively creating metadata of the assigned projects. So, first of all, for each project, participant retrieve the DOI present in the metadata that we provided. They read the publication and extract the necessary information. As a second step, from the publication, they determined if the project was about the human skin microbiome. If it did not, the project was excluded from the skin collection. On the other hand, if it was a human microbiome study, human skin microbiome study, the participants verified if 9 key values found in the publication matched those in the metadata. If discrepancies were found, they added a new column with a standardized name and standardized values. Participants who have created at least 75% of their assigned project received a certificate of participation and academic credit recognition. Additionally, they will be included as co-authors in the publication that will result from this work. Finally, at the conclusion of the workshop on the 15th of May 2024, we submitted a survey to assess the efficacy of our activity using standardized evaluation scales. The survey included 4 sessions that are those indicated here. So the skill of science inquiry, which measures participants' perceived skills, considering both retrospective and current skills, self-efficacy for learning and doing science, which assesses participants' confidence in learning specific science topics and performing scientific activities. And then the last 2 parts of the survey were necessary to evaluate the understanding of the metadata curation process through an open-ended question and to assess their knowledge through true-false questions. So at this point, let me present you the preliminary results from our initial analysis. So at the beginning of our workshop, 29 students attended our lecture, and collectively they were assigned to curate 379 projects metadata, where each of the participants were responsible for a portion of them. At the end of the workshop, 17 students have successfully completed over the 75% of the workshop. For the remaining participants from whom we did not receive any results, we consider them as dropout. We contact them via email, asking them to choose from a multiple choice options to explain the reason why they did not complete the workshop. Out of the 10 answers, 8 of the participants said that the workshop task weren't compatible with their commitments, like study and exams. Regarding the Human Skin Microbiome project, a total of 245 projects were successfully curated. Here are the initial findings based on the 11 survey responses. However, it's important to note that these results are preliminary as they reflect the responses that we received so far. And additionally, due to the limited sample size, precise p-value calculation were not feasible. At the left, you can see the cells plot, which illustrates the average score in confidence in learning in light blue and in doing in blue regarding metadata curation. According to these results, participant scores indicate no significant differences between how they feel about learning and about doing metadata curation. On the right, you can see the SSI plot, which illustrates the perceived skills of our participants before and after the workshop. This plot shows a clear increase in perceived skills following the workshop experience. Finally, I present you the results regarding the true-false score test. In the left plot, you can observe that the majority of participants answers the questions correctly, assuring an accuracy range of 75–100%. However, the remaining participants achieve an accuracy level above the 50%. On the other hand, on the right, we evaluated if the perception of the individuals that I showed you in the previous slide was overrated or underrated, considering the results of the true-false test. As you can see, 73% of the individuals underestimate themselves. So, in conclusion, participants showed increased perception in their learning of metadata curation and ability to apply these skills, indicating effective impact of the data hunters workshop on their knowledge. And our workshop represented the effort to bridge educational and research aims, including students as bioinformaticians of the futures. The workshop outcomes will include the significant educational impacts that will lead to the development of a collaborative curated collection of human skin microbiome metadata, alongside the advancement of bioinformatics dissemination. At this point, I need to thank all the members of my team, the bioresearch team of my lab, the Zooplant lab, and of course, all of you for your kind attention.
Yes. Thank you so much, Sara, for this nice, great presentation. Do we have any questions here in the room? Let's see if our colleagues... Yes, Kath, please.
Really interesting talk. Thank you, Sara. I'm absolutely intrigued by the methodologies that you use to assess the self-competence and the self-confidence and the perception of competence among your students. And it makes me think, well, perhaps in the academic world, more of this is done in research performing institutions, such as my own, the EBI. We don't have a huge amount of experience of doing this, and it's a topic that I think I'd be very interested in learning more about, and maybe something that we could think about doing an ISCB session on in the future. It would be really, really great to sort of gather thoughts on how this, might be done in the future. So thank you for stimulating that thought.
Thank you.
Any other questions or comments? I don't know if do we see something in the chat? No. I had a question as well, but I don't know if there's so if you can elaborate a little bit more on how you have done this analysis of, or how you have evaluated the perception that they had, is something that is more on, it was an open question, it was something that is, how did you analyze that? How did you evaluate that, the perception then of learning?
Yes, the perception, like as the confidence, they are from these standardized evaluative scales. In the slide, I can show you again the slide. So, okay. Okay, I hope you can see.
Yes, we can see that again.
So these are from these publications. The perceptions was about the second one, so is the cell one, and perception in a retrospective. So the skills before the workshop and after the workshop, and they are standardized questions. So there is pre-formulated phrases where you can substitute the object of the question, of the affirmation, because they are affirmation where the students answer from totally disagree or totally agree. So a score of 1 to 1, from 1 to 5.
All right. And I don't know if anyone has another question, because I had another one as well, is how do you imagine extending, so this workshop was for the students, so related to the BICOCA and in metagenomics. Do you imagine extending that to others or to other topics? How much effort would you need for that?
Yes, well, the problem is not, of course, students of BICOCA, because I imagine that it can be extended to all kinds of students, biology students. Indeed, as I told when I say during the bioinformatics lesson, I show the terminal, because it's true. In my course of biology, we don't use the terminal, so it was their first time with the terminal. And however, I see the majority of the efforts in changing, in extending the topic, because it was created on the current metagenomic challenge, which is the lack of standardization in metadata. So it can be used, of course, if there is something else where it's necessary check on publication or another source, and something that is based on step, but from a source that can result in a tabular way. So it's difficult to answer, because yes, now is tailored for a metagenomic problem.
But I can imagine, yeah, extending that also to other areas of metagenomics. So